Okay, hello everyone. I have the pleasure to introduce today Sara Benelli. Will give us a seminar today. Sara is a postdoc position at the moment at the University of Parma. Sara is a benthic ecologist with a strong interest in biochemistry, and among her more or less recent studies, actually the greenhouse gases emission from wetland undergoing environmental restoration. She has also been working on the effect how the benthic coastal buffers against climatic and eutrophication extremes, but also, and most important for us, she's interested in the interaction between sediment communities in aquatic sediment, and in particular about the ecosystem services provided by macrophytes and macrofauna. And I guess that's about this specific thematic that she will talk about today. So thank you, Sarah. It's up to you. Thank you again for giving us this talk.
Thank you very much, Frank. I will share the screen. Can you see my screen? Okay. Thank you very much, Frank, for giving me the opportunity to talk about my research. I entitled the presentation with this biogeochemistry modulated by macrofauna and its interaction with the microbes and primary producers. Oh. Okay. Since the 90s, one of the most engaging areas in ecological research has been the understanding the relationship between biodiversity and ecosystem functioning. Researchers have used different approaches from inferential statistics to detailed analysis of processes and mechanisms, often simplifying experimental setting to isolate specific factors. To me, one of the most captivating aspects in this field has been examining how different species and different functional groups of macrofauna shape benthic biogeochemistry. Although this focus doesn't fully represent the overall impact of biodiversity on ecosystem functioning, it provides a valuable starting point. Observing how these species influence elemental cycles from oxygen and carbon to phosphorus, nitrogen, and so on, open up a world of question for me. And I began as a benthic biogeochemist with a relatively simple experiment that explored this interaction. Each experiment was a stepping stone, raising new questions that led me to modify and expand my approach. What I wanted to show you today is an overview of this journey, highlighting the progression of my work in this fascinating field. In the light of this, I focus my research on benthic compartments of the shallow water ecosystems. Here, the function is governed by the interaction between the physico-chemical environment and the community of micro and macroorganisms. In this environment, exhibits steep redox gradient and complex interfaces, where, for example, fully oxic layers can exist adjacent to sulfidic sediment, as you can see from all these pictures. And what is also benthic fauna through bioturbation contribute to to the horizontal and vertical dynamics and heterogeneity of the sediment. And as I live in a heavily human-impacted area of the Po River Plain in the northern Italy, I'm used to studying eutrophic system. And for this reason, my personal heroes are those very tolerant organisms that do the dirty job. And so they live fully submerged in the organic substrate, but that are capable to change the sediment and so favoring other less tolerant colleagues. And in this presentation, I will explore the roles of bacterial communities, macrofauna, and primary producers in driving benthic processes by analyzing their interaction in a stepwise approach. So I began with simplified experiment setup to isolate and understand the contribution of each functional group of macrofauna that I investigated. And this experiment laid the groundwork for more complex integrated design that revealed dynamics among the different groups. And the central hypothesis of this research is that more complex system, benthic system, enhance resource efficiency by promoting nutrient recycling and, the other end, to minimize the nutrient import and reducing the nutrient loss. So, stepping, no, sorry. Okay, deep diving into the first point, bioturbation studies generally focus on small, abundant organisms, and comparatively, the effect of large organism, organism, and that maybe can be also rare, is less explored. What we found by sampling, it was a canal in the northern Italy that was completely full by a single species of bivalve, as an unionid, very big and large, that was buried into the sediment. And it was compared to most of, not most of, the mussels that are attached to rocks and they are not moving, this mollusk was moving and also a lot into the sediment. By simply attaching a flag on the shell of this mollusk, we confirmed, because we didn't do any experiment to check, but they were moving and moving a lot in this canal. And what we wanted to investigate is about this mollusk is that by means of short-term batch incubation, its role in the regeneration of nutrients. And for this reason, we incubate sediment with the mollusk, and or the mollusk only in the water, to analyze the direct and indirect effect that these mollusks have in the system. And what we found was that is already known, that mollusks regenerate nutrients to the water column by direct excretion, as you can see from this graph, all of the inorganic nutrients. And at the same time, while they contributed significantly to oxygen and carbon dioxide fluxes through their respiration, they indirectly affect or influence the anaerobic metabolism. In fact, as you can see, we measured a very large efflux of methane in the presence of the mollusk into the sediment, that we speculate that it was due to the bioturbation of the mollusk into the sediment, or to do its biodeposition of feces and pseudofeces, that with the reworking it could enhance the anaerobic metabolism inside the sediment, the anoxic sediment. Furthermore, we measure an increase of N2 production Also, in this case, we speculated that it was due to the bioturbation of the mollusk because it was in a eutrophic system, and so this the bioturbation by the mollusk could increase the input of oxic and nitrate-rich water into the sediment that could enhance the denitrification processes. Focusing more on denitrification, there are small and very abundant organisms that can enhance the nitrogen removal by the denitrification. And in this study, we investigate the role of two burrowers. One is superficial burrowers, Corophium insidiosum, and one was a deep burrower in two eutrophic lagoons. And because our hypothesis was that these two different organisms could, with their different ventilation activities, could differently enhance the denitrification. In fact, what we found is that both organisms increase a lot because it's 1,000 micromolar per square meter per hour, the removal of nitrate from the system. The underlying mechanisms are different because the chironomids increase the denitrification associated to the nitrate that is present in the water column because they have intermittent ventilation that enhance the input of nitrate and that is going down deep into the burrows and could be denitrified in the anaerobic niche that are in the close to the burrows. On the other side, the surface burrowers, like Corophium that is an amphipod, it continuously ventilates the burrows, and this allows the ingression not only of ammonium that is excreted and is present in the water column, but also of oxygen. And this combination increases the nitrification process and the associated denitrification to nitrification. Compared to the studies on the nitrogen cycling, what is less explored in the literature is the effect of bioturbators, and in this case of the bioirrigation, on phosphorus cycling. And for this reason, we investigate and we collect one surface and one deep burrowers from the Goro Lagoon in Italy, and that is in a eutrophic lagoon, where these two organisms are present and in high density. So we reconstruct cores to analyze their effect singly and in combination on phosphorus cycling. And what we found by the absolute value of oxygen and dissolving organic fluxes plotted as a function of dissolving organic fluxes for all treatment show no significant difference between the benthic respiration and the phosphoregeneration within the treatments. But what we found is that the ratio between two processes is constant, meaning that there is no direct effect of the bioturbators on phosphorus fluxes. What we found by to understand more the why this phosphorus was not so different between the different functional groups, we incubate the two burrowers in the water to measure the excretion rates. And what we found is that the amphipods were excreting directly the phosphorus into the water column, but this was not true for the polychaetes. And we didn't measure what was in the feces, in the pseudofeces of these two organisms, but we analyzed the pore water from the different treatments. And the observed increase of phosphorus in the presence of the polychaetes, we thought it was an indirect due, for example, to its bioirrigation, more than bioturbation, deep into the sediment. Because if you see the fluxes of the profile of phosphate in this among the treatments, you can see that the concentration of phosphate is much more when the two organisms are not present, suggesting that this activity of bioturbation can increase the mineralization rates of phosphate, for example, by particle reworking, and increase the aerobic microbial activity. Furthermore, they decrease also the presence of reduced metals, suggesting that their bioturbation increase their oxidation of the sediment, as you can see, for example, from the profile of the redox potential. We also measure the pool of ferric iron, and here I presented the percentage, and as you can see, the control has the lowest percentage of ferric iron compared to the other treatments. Since this kind of studies only reflects the dark part of a daily rate, we then move to the next set of experiment will shed light on the interaction among macrofauna, primary producer, and microbes in light-exposed sediments. I moved to the north of Europe for these experiments, and in particular to Lithuania. And here they have the largest lagoon in Europe, and it is again eutrophic. So I continue my path in the eutrophic environment. And here I investigated the role of very abundant animal, the chironomid larvae, and its effect with the microalgae that were naturally growing on the surface sediment. We to measure their effect, their role on the benthic biogeochemistry, we reconstruct cores with sediment, organic rich sediment, and we add different density of the chironomid larvae, a low and high density. What we found is that generally the sediment with and without the chironomids were sink for nitrogen and for phosphate on a daily basis, as you can see from the dots. However, in the dark, in the night, there was always a regeneration of phosphate regardless of the presence of the chironomids. And by analyzing also the pore water of this sediment, we find interesting results because, as you can see from the bottom graphs, the presence of the chironomids almost set to zero the regeneration of the nutrients, as we saw from the net fluxes before. And this can be due to different mechanisms, of course, depending on the element. For example, we speculated that the decrease in ammonium fluxes was due to the increase of denitrification, coupled to nitrification, because their borrowers, they're changing the particles and they're ventilating the burrows, introduce into the sediment oxic water and again nitrate rich water, so could enhance the denitrification. For phosphate, instead, the presence and the injection of oxygen into the sediment via the bioirrigation increase the co-precipitation of phosphate with the iron as a net effect. And other, of course, processes that we didn't measure could be the reduction of the increase of the aerobic mineralization in the presence of the chironomid. And the net effect that we saw is that there is a lower regeneration of the nutrients to the water column due to the combined action of the microalgae and the chironomids. However, as you can see from the literature, there is there is also an effect as these are larvae of insects that then fly out from the sediment. What happens when they fly out? What is the burrow? The burrow is not anymore oxidized by the ventilation of the chironomids, and so all the nutrients and the trap like phosphate, for example, that were inside the sediment, what happens when they fly out? And so this is still an open question that we didn't explore, but of course is very interesting. And other groups of macrofauna can have a very different effect on on retention and regeneration of nutrients. In the same lagoon, we sample a shallow area that was covered, or at least there was a high density of these two mussels. Was an unionid that was most of the time covered and colonized by dreissenids. And for this reason, we decided to understand the role of the native unionids and invasive dreissenids in this lagoon. By collecting intact core with the different mollusks, because they were visible by eye due to the low depth of the sites, we measure the effect that they have on the regeneration of nutrients. And so by means of light and dark incubation, we found that, for example, for ammonium, there was always a regeneration of ammonium by direct excretion of the mollusk. While during the light, the effect of the microalgae and the phytoplankton there was in the water was higher. For silicate, instead, the sediment, regardless the presence of the mollusk, were always sinks, suggesting that in this period the presence of diatoms were higher. And for phosphate, it was a nice result because it was the only one that was different between the two mussels. And what we know also from the literature is that the dreissenids have a huge contribution and a huge role in regenerating phosphate. In fact, it was both in light and dark condition was always regenerated. And to deepen the role of dreissenids, we did also another experiment with the same mussels, in which we also evaluated their role in if they enhance the phytoplankton growth, the phytoplankton growth rate. In fact, we with the use of dialysis bags, we incubate again the four condition just in the water, in situ water, to measure if the presence of the mollusk and how they increase the phytoplankton rate. And what we found is that there is a net effect and a significant effect of dreissenids on the phytoplankton growth. And I will not show, but also this means that the Dersanis decrease the N2P ratio, so nitrogen to phosphorus ratio. And this has an implication, and very high implication, on the blooms, for example, of cyanobacteria. This is a picture that we took in the Curonian Lagoon. This is a very big problem in this lagoon. The interaction between macrofauna and primary producer is not only related to microphytobenthos or phytoplankton, but can also occur between burrowers and macrofauna and rooted plant. In this work that I show, by sampling, we find very often these worms, that is an oligochaete, that is inside or in the rhizosphere of a rooted plant. This is Vallisneria spiralis. The characteristic of this plant is that it can release oxygen via the roots and oxidize the sediment around the roots. What is we studied a little bit more about this worm, and we found that it can respire oxygen through the skin. And so this was an explanation why we always find, or most of the time found, this association in the field. And so we decided to investigate also the role of this combination along the physico-chemical cycling. And what I want to show you today is the effect that the combination singly and together have on methane evolution. So compared to the other experiment that we had the short-term batch incubation. In this case, we incubated the reconstructed sediment for 12 days, and we put a reverse funnel on the top to trap the bubbles that were coming from the sediment in different conditions. And what we found in the 12 days of incubation of this microcosm is that the combination of the worms with the macrophyte decreased by 97% the ebullition of methane. It is huge compared to what can be released just in the sediment. And of course, what can have happened in this case is that the plants, since we measure that the 30% of the photosynthetic oxygen that is produced by the plant is released from the roots to the surrounding sediment, it can, of course, oxidize the methane that is present and with a high concentration in this sediment, as it's very organic. And at the same time, the worms, with its bioturbation, with its ventilation activities, can introduce oxygen and nitrate that can inhibit the production of methane. And it's released to the water column. In the final section of this presentation, I want to focus on the relationship between the complexity and the functioning of this benthic compartment, and in particular along environmental gradient. At first, I investigated the role that a deep burrowing oligochaete have on nitrogen cycling. And this invasive worm was present in two very different environment. One that is eutrophic with high nitrate concentration and very high organic matter content. And one oligotrophic that has the opposite characteristics because it has very low concentration of nitrate and low organic matter content. What we found in this with this study is that the denitrification was measured and was measured in both sides, and as we added to in that course increased number of the oligokits, the denitrification was increasing with the biomass of the worm. And of course, it was much higher in the eutrophic side compared to the oligotrophic side due to the higher nitrate concentration in the water column. In fact, I also reported the fluxes of the nitrogen nutrients, and you can see that in the eutrophic site, ammonium is the white bars increase with the increasing of the biomass in both sides. However, there was a nitrate, negative nitrate fluxes in the eutrophic site due to the high concentration that we have in the water. And what is different between these two is that in the oligotrophic site, there was, when there was no biomass and the lowest biomass, the sediment after sinks of nutrients, nitrogen nutrients, while when they increase, also the regeneration of nutrients increase. What does it mean this? We measure the denitrification efficiency that represented the fraction of mineralized nitrogen is released to the water column as N2. And as you can see from the graph, it was from 60 to 80%. So almost all of the nitrogen that was present in this site was denitrified. While, for example, instead for the oligotrophic site, there was efficiencies very high with no biomass and with the lowest biomass, but it decreased. What does it mean? It decreased because the regeneration of nutrient in this case was higher than the N2 production. And this is to show that the same organism, the invasive in both places, can have a very different effect. Maybe a positive effect in the eutrophic site, since the eutrophic has a lot of nutrients, and if the nitrification is high, is very good for the system. For the oligotrophic sites, this organism is not good from a point of view of nutrient regeneration, as the recycling is much more higher than the denitrification. The last study that I want to show is in the same study site. I investigated the role of another invasive organism that is a clam, and the rooted macrophytes, because in the most of this the sampling that we did in these sites, this clam was always close to the middles of these plants, and the plants, both plants, are rooted submerged macrophyte. Valisneria spiralis is a plant that is has high plasticity. So we found in very different and heterogeneous places. So from eutrophic to oligotrophic, while Lobelia dortmanna is a very specific plant that is can be found just in specific site, oligotrophic with the determined characteristics. But lobelia, compared to Vallisnera, can release 85% of its oxygen that it was produced by photosynthesis via the roots into the sediment. So it has an incredible characteristics. For this experiment, we collect the cores from the two sites, and what I reported here is I focus on denitrification. And what you can see from these two graphs is that in both sites, in the four treatments, so only sediment with the macrophyte, sediment with the mollusk, the clam, and they both together. For the eutrophic and for the oligotrophic site, is that the N2 production was higher in the eutrophic site due to the higher availability of nitrate, and was always present in the four conditions. What is very different and also sometimes somehow surprising is the light results. In the eutrophic site, we found that with the macrophyte, nitrogen fixation, so why these are negative fluxes, was higher compared to the condition without the macrophyte. And this could be explained by the fact that the plant assimilate ammonium from the pore water, and so it needs more ammonium for its growth. And compared to macrophyte alone, when was present the mollusk, the nitrogen import was less, suggesting that the recycling of ammonium by the mollusk and its excretion help the macrophytes. For the eutrophic site, we have another result. We found much higher nitrogen fixation in the condition with the sediment and the sediment with the mollusk. While with the plants, there were much lower nitrogen fixation. And this could be explained by two reasons. One is that this oxygen that is released from the plants inhibit the nitrogen fixation. And the second is that in this condition, there was a huge negative fluxes of ammonium, meaning that there was a high availability of ammonium in these two conditions. Ammonium and the oxygen can increase the nitrification, and the nitrification increase the denitrification. So the net effect that we found is a less negative fluxes with this plant. To conclude with something that can be added for the next step, a colleague of mine use the experimental data that we had to model and to use to apply a modeling approach. In this study, we analyze the effect of two bioturbators, filter feeders and deposit feeders, and two growth form of primary producers, the rooted macrophyte and the microphytobenthos, on the benthic nitrogen cycling in a eutrophic system. With the ecological network analysis, we could understand and estimate the indirect effect that this interaction have and that we couldn't measure experimentally. In fact, we found that a major fraction of nitrogen that was assimilated by the macrophytes was processed by macrofauna, even if there is no direct link between the two compartments. And in other words, this application demonstrated the dependency of the primary producers from the particulate nitrogen that was facilitating by the presence of the macrofauna. So this is an example of the next step or this application of a modeling tool to an experiment at the micro scale. And of course, there can be interesting point also to identify with molecular tools that are much more fashion now nowadays if there are interaction between the host and the microbes occurring between what we found, or at least visually found between the primary producers and the macrofauna. And so with this, I thank you all and thank you for listening to me and to my research.
Thank you, Sarah.
Thank you.
Thank you for your talk. If anyone has any question.
I have one to start, if that's okay. So I thought your results with the oligotrophic, getting very different results between the oligotrophic and eutrophic systems are interesting. And I was wondering if you've thought about feedbacks and whether the critters are kind of, in the like what do the impacts do in terms of changing the systems over time, or do you think that there's an impact? Like, are there positive feedbacks that maintain eutrophic conditions or vice versa? Does that make sense?
Yes. Of course, there are multiple feedbacks because in the oligotrophic side, the regeneration, of course, can be negative if it's maybe increasing and changing the primary producer dominance from rooted macrophyte to phytoplankton, this shift. But at the same time, it's a way for the plants to live because they need anyway nutrients for their living. So it's difficult to say if it's a good or a bad feedback, this presence. So it's it's invasive, not always is a bad thing. I would say. I don't know if I answer.
Thanks.
Any question? So I ask a question. Sarah, okay, here we were mostly talking about ventilation, irrigation, and stuff like this. I just have a question about the unionids and the dracenids stuff. The unionids, they're supposed to burrow and to move the surface of the sediment. And do they do the same when they are covered by dracenids?
Mhm. Yes, what we found, it was always the unionid that was inside and the dracenids up. So the dracenids were out of the sediment, but the unionids were always digging or try to dig into the sediment. Yes, even if they were covered from the other mollusk.
Okay, that means that the volume of the sediment that can be moved by the unionist is not higher when when they have dressing it because they don't burrow. You have the
Exactly.
dressing. Okay.
We didn't find the dressing inside. No, never. Maybe it's they cannot only because they
Okay.
are.
So that means that's one. The onionists they were playing their role on both the sediment on the surface and the sediment only in the water.
Yes.
I think. Okay.
Yes. Yes.
Okay.
So I have one more. If that's I don't want to. So looking at the chironomid data, it looks like you've the abundance is high enough that basically they're totally flushing the pore water. And I'm wondering if you have a sense of what the threshold is, like if you have a small enough number of them, do you have something in the middle of your control and your experimental, and do you have a sense of like what that threshold is? Like how many worms do you need to have in order to have a fully flushed upper layer of sediment?
Okay. Well, I didn't show, but there was also the part with the low abundance in this experiment, and it was like half of the other two. So I would say that the abundance or the density that we added is like in situ density that are found in the Coronal Lagoon. But I don't know if more than they can do differently, but the low density was exactly in the middle between the control and the high density. So I don't know if after this, if you add more, the threshold can change because then there is also other factors that can play. But it's yeah, I think it's
Fairly additive.
seems yes.
People are shy tonight. This afternoon. Tonight.
It's,
It is. Okay. Then I have a question. The alien swarms. You did the experiment in eutrophics and oligotrophics condition. Do they invade the same those two sites in natural way? I mean the concentration. Sorry, the density you use of worms, will they be the same in both lakes, by example, in the eutrophics and the oligotrophic in the natural way?
well, good question. I don't know. I don't know exactly the density in these two sites, but it was easy to find. So we added realistic abundance, but I don't know if there are more because the environment that we sample it was like a part but of a very big lake, and so I don't know if in the other places the density can be different, but it was in both cases was realistic of the site. We didn't add more than to enhance the processes, or it was realistic.
Okay, thank you, Sarah.
Thank you.
I have a question about the methane emissions. And I was wondering, you did experiments that were 10 days long. I was wondering, so in the natural wetlands, oftentimes methane emissions increase when you have vegetation because of the capillary action. The roots are transporting dissolved methane from that's produced below the roots or maybe between the roots and into the atmosphere. Given that these experiments were 10 days long, do you think the results would change if you had done this for, let's say, a year or allowed for this full establishment, or maybe I'm misunderstanding something?
It's right what you said. I think it depends more on the typology of the plants, because in this case it was a rooted plant submerged, and it has no like the parenchyma or the canal that are going up to the atmosphere directly. So in this case, its effect is more on the roots, on the sediments, than on the water column, for example. And of course, if you have plants that have just a canal from the sediment to the atmosphere, in this case, it can be very different. And the effect, I didn't test, but I think would be the same. The increase that we had at the beginning is because we reconstructed the sediment, and so at the beginning there is this big enhancement of methane ebullition, but then when it stabilizes in the days, it start to release, but with another trend. And I think the effect would not be different if you keep keep taking this experiment with this plant. I think, yeah.
And do you think it's mainly suppression of methane production because of oxygen intrusion, or could there also be a stimulation of methane oxidation by either the worms also, because they also were lowering the methane emissions, that you have increased iron oxides, maybe not necessarily, or oxygen, and then methane oxidation in those deeper sediment layers that prevent the methane from bubbling out or diffusing out into the overlying water.
Yeah, this is a speculation because we didn't test anything, but of course, I think with molecular technique, for example, just to see the community of the bacteria around the both organisms in the different could help a lot what we can do experimentally, and it's a very good point. Yes.
Thank you.
Okay. So Sarah, looks like that we don't have any more questions. Thanks again for accepting to give us your interesting talk. Also to agree to the talk to be recorded. I received a lot of messages during the talk that people they couldn't join because I sent a recall and they couldn't join a meeting or whatever. So I will be happy to have it on the YouTube channel. And so you're the last one of this year. And just tell the people that with Adrian and Kelly, we will start soon to prospecting to propose you as nice seminars as the one we had this year and the one you completing today this series. Sarah, thank you again for that. Thanks for all of us for attending, and see you soon. Bye,
Thank
everyone.
you.
Thank you again, Sarah.
Thank you very much. Thank you to you. Bye.